Hey guys! We're Group 11 from Bio 2.
My name's Daniel. I will be talking about transcription in regards to initiation, elongation, and termination in prokaryotes.
I'm Rebecca, and I'm going to be talking the translation of prokaryotes, which will include initiation, elongation, and termination.
And I'm Daisy. I'll be talking about antibiotics in both eukaryotes and prokaryotes.
I'm Marissa, and I'm going to be talking about initiation in eukaryotes in both translation and transcription.
I'm Esme, and I'm going to be talking about elongation for translation and transcription for prokaryotes, as well as the post-transcriptional modifications for mRNA.
I'm Claudia. I'm going to be talking about termination in both eukaryotic translation and eukaryotic transcription.
Hello everyone, my name is Daniel Navarro and I will be teaching you guys transcription in prokaryotes. For starters, it requires the DNA double helix to partially unwind in the region of the RNA synthesis, which I will be explaining in a little bit. The region of the unwinding is called a transcription bubble, which is basically in a bubble all around this strand over here. Transcription always proceeds from the same DNA strand for each gene, which is also called the template strand, as I have down here, as you can see. And the template strand goes from the 3' to the 5' end in the right direction. And the RNA product is complementary to the template strand and is almost identical to the other non-template strand, the DNA strand, also called the sense or coding strand, which as you can see is above the template strand. And it goes from the 5' to the 3'. And as you guys can see that both of these strands start in opposite directions of the numbers. So 5', this starts at 3'. As you can see that. The only difference in that is that in RNA, all of the T nucleotides right here are replaced with U nucleotides. The nucleotide pair in the DNA double helix is that it corresponds to the site from which the first 5' mRNA nucleotide is transcribed is called the +1 site or the initiation site. Nucleotides preceding the initiation site are given negative numbers, as you can see here down below, the -35 bases and the -10 bases. Conservatively, nucleotides following the initiation site are denoted with the plus numbering and also called downstream nucleotides. A promoter is a DNA sequence. And as you can see in this figure right here, the promoter is from this area to this area, the main region of the template strand or the strand. A promoter is a DNA sequence onto which the transcription machinery binds and initiates transcription. So it starts transcription. In most cases, promoters exist upstream of the genes they regulate. The specific sequence of a promoter is very important because it determines whether the corresponding gene is transcribed all the time. Some of the time or infrequently.
Good morning, afternoon, night, whenever you're watching this. I don't know when you're watching this, but you're watching it. So we're going to talk about the translation of prokaryotes. Not eukaryotes, you'll know the difference of that by now, hopefully. So basically, I'm going to start by talking about initiation, and then I'm going to go into elongation, and then we're going to end with termination. Ironic, right? So before we begin, I'm just going to tell you about the parts that happen in translation of prokaryotes. So the first part is the anticodon, which is the opposite of the start codon. As you can see, it has the A's, the U's, and all the nice stuff. And then we have the tRNA with the first amino acid as well. Then we have the large ribosomal subunit, the small ribosomal subunit, the mRNA, the start codon, which is on the 5' end, and the stop codon, which is on the 3' end. So to begin, I'm going to talk about initiation. Basically, before I'm going to give you a little bit of background of initiation. The bacteria has several varieties of a sigma factor. Each in itself has expressed itself under different environmental conditions. In this way, the different combinations of the genes can be transcribed. And basically, that's the beginning. And then it's very fast-paced, and now we'll get into what initiation is. So in initiation, it's when the translational complex is formed, as you can see here. and the tRNA brings the first amino acid in the polypeptide chain, which is right here, to bind the start codon onto the mRNA, which is right here, and this is the start codon. And then we have elongation. In elongation, it's basically a term that's used once the RNA polymerase enzyme has begun movement along the gene. And it goes very fast. The pace is at 1,000 bases, per second. And basically what elongation is, the tRNAs bring amino acids one by one, as you've seen in the previous step right here. And then it makes a polypeptide chain. And then once a polypeptide chain is formed, one by one, these are knocked out for each amino acid that is added. And then we have termination. And termination is just when this is the end of everything, basically. It's when it's going to be terminated, as you can see in the word termination. So the release factor, which you can see right here, recognizes the stop codon, which is right here, and then the translation complex dissociates, and completed polypeptide is released. And here's your completed polypeptide, and then this one's almost completed, and then the release factor. And then once all that's done, the components are recycled and ready to start again in initiation.
I will be talking about the initiation and transcription of eukaryotes. Initiation in eukaryotes is the main point from which the cell selects which proteins or RNA is going to be produced. The RNA polymerase, which is located right here, must be able to recognize the start of the gene and bind firmly onto it and its transcription start site. In eukaryotes, there are 3 different types of RNA polymerases. RNA polymerases 1 and 2 transcribe the genes encoding transfer RNA, ribosomal RNA, and other various RNAs that contribute to structural and catalytic roles of the cell. Unlike in prokaryotic cells, eukaryotes require several other proteins called transcription factors, which are located right here, to first bind to the promoter region and then decide which polymerase to use at that point. The most important core promoter element in eukaryotes is known as the Tata box, which is found 25 to 30 base pairs upstream from the start site of the transcription site. The Tata box is in the binding site for a transcription factor known as the Tata binding protein, otherwise known as TBP, which is a subunit of another transcription factor, which is transcription factor 2D, otherwise known as TFIID. After TFIID binds to the Tata box via the TBD, 5 more transcription factors and RNA polymerases combine around the Tata box in a series of stages to form a pre-initiation complex, which is this. This diagram demonstrates the process of initiation in eukaryotic cells in transcription. As you can see, the Tata box, which is placed in the promoter region, which is located right here, is also where the general transcription factors are located, which are right here. The Tata box is categorized as a general transcription factor, which bind to the promoter region to decide which RNA polymerase will be used, as you can see in the diagram right here. On top of the RNA polymerase, these 3 dots right here are the post-transcriptional RNA modification factors. These factors will be described later on in the video. But once the initiation process is completed and the polymerase is selected, the transcription process continues and the RNA strand is produced, which is right here, and it reads from the 3' end to the 5' end of the DNA strand.
During elongation of eukaryotic transcription, the rRNA begins synthesizing a complementary RNA strand to the desired gene in the DNA. It starts at the DNA sequence TAC, which when transcribed into RNA is AUG. AUG is the codon for methionine, which is the start codon for all mRNA transcripts. It creates the RNA polymerase 2 synthesizes its RNA strand by complementing each of the DNA nucleotides. The only exception when it comes to the complementary nucleotides is instead of thymine, adenine is paired with uracil for mRNA. The RNA polymerase begins synthesizing a complementary RNA strand to the desired gene. This RNA strand will eventually become translated. RNA polymerase 2 begins transcripting the RNA strand at the nucleotide sequence TAC. When transcribed into RNA, it is AUG, which is the codon for methionine, the start codon for all mRNA. It continues synthesizing the mRNA strand from the 5' to the 3', in the 5' to the 3' direction. It complements the current strand of DNA with a paired nucleotide, with the exception of thymine. With the exception of thymine. Instead of the A's being paired with thymine in the nucleotide strand, in the RNA strand, thymine is replaced with uracil.
Now we're going to be talking about eukaryotic transcription, specifically termination. So the termination of transcription is triggered by a termination sequence. This process begins when specific base pair sequences from the ribosomal RNA or rRNA are recognized by a termination protein called TTF1, also known as transcription termination factor for RNA polymerase 1. These recognized rRNAs were transcribed by RNA polymerase 1, which then causes the termination factor to be used specifically for this polymerase. TTF1, or the transcription termination factor for RNA polymerase 1, however you want to reference it, is able to bind DNA at the recognition sequence and prevent any further transcription. After the message that the transcription process is finished is received, RNA polymerase 1 detaches itself from the DNA template and liberates the newly synthesized strand of RNA. Although the signal to terminate transcription may be at a certain pinpoint location on the strand, RNA polymerase 2 continues to transcribe the RNA from a range of a few bases to a few thousand base pairs after the signal. This is because the regulatory RNA genes deciphered by said RNA polymerase 2 lack the ability to terminate the function of the polymerase at any specific location on the sequence. While RNA polymerase 2 continues to transcribe the strand, the transcript can be cleaved. When this occurs, the upstream portion can be released, functioning as pre-mRNA. The remaining surplus of genetic material is then digested by a 5' exonuclease, the enzyme that removes extra nucleotides from the 5' end of a polynucleotide molecule. While the cleaving process is happening, RNA polymerase 2, the endonuclease, helps liberate the polymerase from the strand. After the RNA polymerase is released, the strand can be capped at the 5' end, terminating the transcription in that section of the gene. And the cap is a 7-methylguanine cap, and on the 3' end of the strand, the sequence gains a poly-A tail through the process of polyadenylation. This is just when adenine is added to the strand. The addition of the poly-A tail via polyadenylation helps mature the mRNA for the next step, which is translation.
Unlike prokaryotes, eukaryotes have a nucleus, and once our DNA is translated into mRNA transcripts, it's still within the nucleus. Our RNA subunits and other RNAs and other proteins are within the cytosol. Regular mRNA transcripts are unable to survive in the cytosol because hydrolytic enzymes break down the phosphodiester bonds between the nucleotides. So in order for the mRNA to mature and be able to survive outside the nucleus, it needs to gain a 5' cap and a poly-A tail. The 5' cap is comprised of a triphosphate bridge with a 7-methyl guanine cap. This is on the 5' end. This cap also indicates where the start of the transcription once it's within the RNA subunits. On the 3' end, we have a poly-A tail. This is created via polyadenylation, and it is when between 150 to 250 adenine nucleotides are added to the 3' end of the mRNA. Housed in between these boundaries is our mRNA, our coding sequence. However, within our coding sequence, we have multiple exons and introns. Exons express the proteins that we are trying to create, and they are the coding sequences that we want. Lastly, the spliceosomes must remove the inexpressive introns that are riddled within the exons. This leaves the protein coding regions called exons. Since 98% of our DNA is full of these non-protein coding regions that live within the protein coding regions, the last step of post-transcriptional mRNA modification is splicing out the introns from the exons. Introns are non-protein coding regions that are inexpressive because they are junk DNA. They are the transcripts of junk DNA. Junk DNA is riddled within our protein coding regions so that in the event of a mutation, it is less likely for it to occur within a protein coding region. Should a mutation occur within a protein coding region, the effects could be harmful once the proteins have been created. So the first thing that occurs for the spliceosomes for the introns to be spliced is a SNRP called U1 binds to the start of the intron, while a SNRP called U2 binds to the end. After the binding of additional SNRPs in these such as U4, U6, U5, and U4, U6, and U5, we then have the assembly of the spliceosome. Next, the laureate loop is formed by the esterification of a guanine on the start of the intron, binding to the hydroxyl group of the adenine at the binding site. At the branch, a laureate loop is formed by the binding of a guanine to the hydroxyl group of an adenine located at the bridge site. This then splices. This then causes the spliceosome to cleave at the E2 site, and a laureate loop is formed, which then allows the two exons to bind together. The spliceosomes are removed and allowed and recycled for later use.
In initiation for translation in eukaryotes, a ribosome, which is made up of a ribosomal large subunit and a ribosomal small subunit, and an mRNA with instructions for the protein that will be made, and an indicator tRNA carrying the first amino acid in the protein, which is almost always going to be methionine, or MET for short, bind together to form the initiation complex. First, shown in the diagram, the small ribosomal subunit and the initiator tRNA bind to the 5' cap of the RNA, and then after that, the complex made up of the tRNA and the amino acid in the small ribosomal subunit slide across until it finds the start codon, which is almost always AUG. And then once it finds the start codon and binds to it, the large ribosomal subunit, which is pictured right here, joins to form the initiation complex, which is this whole thing right here.
Eukaryotic translation elongation is a 4-step cycle that continues until the protein it is creating is completed. The ribosomal subunit created that reads the mRNA has 3 sites: the exit site, E, the peptide site, P, and the aminocidal site, A. Note, only 2 sites are ever housed at the same time. Here in the first step of our process, we have a growing peptide chain that is attached to this third tRNA. We have a new incoming tRNA, number 4, that matches its anticodon with the codon present in the mRNA strand. This incoming tRNA brings with it an amino acid that forms a peptide bond with the growing chain. Here, once the bond is created with the chain, the amino acid is no longer attached to its tRNA. Since the tRNA no longer holds its amino acid, the large ribosomal subunit translocates to the right as a means to shift the tRNA forward into the exit site for it to be ejected. The small ribosomal subunit translocates also to the right, and this forces the tRNA to be ejected. This tRNA can be reused once it's been reattached with an amino acid. The elongation cycle continues once a new tRNA binds its anticodon with the next subsequent codon in the mRNA strand.
So now we're going to be talking about eukaryotic translation, specifically termination. Termination is similar in eukaryotes as it is in prokaryotes. The only difference involves the release factors. So termination of translation is initiated by stop codons in the mRNA. These stop codons include UAA, UAG, and UGA. These sequences on the mRNA are recognized by release factors. The 2 release factors involved in eukaryotic translation include ERF1 and and ERF3. ERF1 recognizes all 3 stop codons. ERF3 is a GT pace that helps ERF1 release the completed polypeptide. So the release factors are able to manipulate the enzymes that form the polypeptide bonds on the strand. These enzymes are tricked to add water to the molecule's last amino acid on the chain, and because that part of the chain is hydrophobic, the sequence separates itself from the transfer RNA, or as you know, tRNA, surrendering the newly translated protein, and once translation is terminated, the molecule can go on for another round of translation if it wishes.
Okay, so today I'll be talking about how antibiotics works. So antibiotics first originated in fungi. The function of antibiotics is they attack bacteria this way. They do it by attacking the cell wall. So they poke little holes into the cell wall of the bacteria, and this allows the bacteria to expand and eventually pop. So there's different types of antibiotics, such as the book talks about these ones. Tetracycline, streptomycin, chloroampicillin, cyclohexamide, and rifamycin. So these all have different functions, and this is the chemical structure for tetracycline. We're going to focus a little bit more on this just because I wanted to get it really in depth, just to have one example. So these are all bacterial protein inhibitors, or they also inhibit RNA synthesis. So the different types of bacterial protein inhibitors or RNA synthesis inhibitors the book talks about tetracycline, streptomycin, chloroampicillin, cyclohexamide, and rifamycin. So these all have different functions. Today we're going to mostly focus on the tetracycline, and this is the chemical structure for it, and it's kind of like the same. So this is just like kind of like a broad type, because there's like more specific ones. Like these antibiotics can be taken in dosage high enough to kill bacteria without being toxic to humans. Because different antibiotics bind to different regions of the bacterial ribosomes, these drugs often inhibit different steps of the protein synthesis. So then this means that it can affect the RNA synthesis or different mechanisms, like different antibiotics target different regions. So then the book mentions these antibiotics specifically, but there's a lot more, of course. And there are many types of antibiotics, and they were first isolated by fungi. So fungi and bacteria often occupy the same ecological niches. So because fungi and humans are both eukaryotics and are thus more closely related to each other than to bacteria, we have kind of borrowed some of these mechanisms to fight against bacteria. Eukaryotic cell has the 50S subunit and the 30S subunit, while the eukaryotic has the 60S subunit and also the 40S subunit. And so the way that tetracycline goes in is that it only binds to the 30S subunit. So the eukaryotic cell will remain unharmed since that's a part of the organism. We only want to attack the bacterial cell. So then the tetracycline, it goes into the mouth of the 30S subunit, and it from getting inside. So then this means that the tRNA can't bring the amino acid, which then means that the bacteria is unable to grow. Thus, the tetracycline's job as an antibiotic is complete.